Hi everyone, welcome back to Useful Genetics. This is Lecture 1I, where we're going to talk about the fundamentals of what chromosomes are. We'll talk about both their physical structure and their informational content. We'll talk about the regulatory signals that a DNA molecule needs if it's to function as a chromosome. We'll talk about the different versions and the terminology that we use for these, and we'll talk about how we can represent chromosomes. Now, structurally, a chromosome is one very long molecule of DNA. Human chromosome X is 150 million base pairs long, very long. And the DNA isn't naked in the cell. The DNA is bound to and wrapped around proteins, as we described earlier. Informationally, a chromosome is one very long DNA sequence. And embedded in this sequence are sequences that specify genes and other functions, and they're embedded in a background of non-functional DNA sequences. The diagram on the right is various representations of aspects of the human X chromosome. We'll come back to representation later. Now, if a molecule of DNA is going to function as a chromosome, it has to have particular properties. It has to carry specific information. First, it has to have signals that are represented by DNA replication proteins. These signals are called origins of DNA replication, and there's usually multiple origins of replication along the length of a chromosome. Chromosomes also need special sequences for where DNA replication ends. At the ends of the chromosomes, there are special sequences called telomeres. And these sequences exist because the ends of DNA molecules are harder to replicate than the internal parts of DNA molecules. Chromosomes also have to have special attachment point sequences called centromeres, one to each chromosome, located at a particular place on the chromosome. And this is the place that fibers attach to when the cell is going to divide to pull the chromosomes apart. Finally, of course, chromosomes have to have genes. We'll talk about the genes on chromosomes in the next lecture. Now, human chromosomes, luckily, are fairly typical. We're fairly ordinary animals genetically. So we have, if you're female, you have 23 different chromosomes. If you're male, you actually have 24. Our chromosomes, that's different chromosomes. You have two versions of each of these chromosomes, except for the X and Y chromosome if you're male. And we'll talk about this in Module 4, I think. Now, each chromosome, again, the X chromosome I described was typical. Each chromosome has got between 50 and 250 million base pairs of DNA. That's how long it is. And in that DNA sequence is the information for between about 400 to 4,000 genes, depending on the chromosome. The longer chromosomes have more genes on average. And as I said, each chromosome has different genes. Not just different versions, but completely different genes on each chromosome. Now, not all organisms have, modest numbers between, say, 5 and 50 or so chromosomes. There are some organisms that have all their genes spread over only a very few chromosomes. Some organisms will have their genes spread out over hundreds of tiny chromosomes. Now, these organisms generally have similar amounts of DNA to us. It's just that they've spread their DNA out in more pieces. Now, I have to introduce now some very important terminology that it will be critical that you be able to use clearly. And that is two alternative words that geneticists use instead of the word gene, which we've been using gene a lot, but actually it can be fairly ambiguous in important situations. And so geneticists have introduced two other terms, locus and allele. And we need these terms because genes come in different versions. So allele refers to one of the versions of a gene. or a version of a DNA sequence, even if it's not a gene. Whereas locus refers to, it comes from the Greek or Latin meaning place, it refers to the location, the position where the gene occurs, the sequence that encodes the gene on a chromosome. And the term locus is used when you want to discuss the gene as a general thing and to include in your discussion all of the versions of the gene. This applies. So we could talk about the locus that codes for the ability to taste a particular chemical, for instance. And then we might say that this locus encodes a protein that comes in different versions. This locus has different alleles, different versions of the DNA sequence. So allele, where you would refer to version, and locus, where you're referring to the general gene in all its versions. Now, you've already seen several representations of chromosomes, and I haven't really explained them very much at all. So I'm going to talk quite a bit about this in the next couple of slides. So first, here's a representation showing what chromosomes actually look like under a microscope in a cell that was sort of frozen, stopped in the middle of cell division. Now, the chromosomes aren't neatly arranged like this and numbered in the cell. They're all spread out in a bit of a mess. And what the cytogeneticist who took this photograph has done is actually cut out the picture of each chromosome, cut out each chromosome from the picture, and then rearrange them on a piece of paper so that the two versions of chromosome 1 are together, the two versions of chromosome 2 are together, etc. This was the chromosomes of a male because they've got one version of the X and one version of the Y. Now, you notice that the chromosomes are actually, they're not very informative. They just look like little blobs. And sort of they're darker in some bits than others. These darker and lighter parts reflect different properties of binding to a dye that's used to color the chromosomes under the microscope. And the dark and light parts have come to be used as landmarks. So chromosomes are often represented by diagrams that show the dark and light bands as landmarks along the length of the DNA. Now, it's important to realize that these dark and light bands do not represent genes. There are many more genes than there are dark and light bands. They're just staining patterns that serve as landmarks. In the same way that the position of the centromere serves as a landmark. Now, teachers and students often represent chromosomes as sort of fat, blobby X's or skinny butterflies, or they'll just draw a pair of lines like that, like an X, and say, oh, there's a chromosome. That's not how chromosomes look, and it's best that you not represent them as that, because it will actually create confusion in your mind. Now, how I'm going to represent chromosomes, quite a few different ways, but they're all going to be very simple. I might show them as sort of rounded lines like this with constrictions showing the locations of the centromeres. It's not that the length of the position of the centromere is that important to what I'm talking about, but because it differs from chromosome to chromosome, they serve as ways to reinforce the point that these are different chromosomes with different genetic information. I've also drawn them different lengths to emphasize that point. More simply, I could draw chromosomes just as lines. This is especially likely if I'm just drawing them freehand. I might draw a line, and then I might draw a blob on it to indicate that this is the location of the centromere. I might sometimes just draw the lines without the centromeres. I may draw them as wiggly lines, especially if I want to emphasize that they're not neatly arranged in the cell, but they're actually sort of randomly located around the cell. I may draw them as I've done here, where I'm showing all of the chromosomes from a particular person in the same color. And of course, I haven't drawn all 23. I've only drawn a small number because that's all I need to make the point. And so I've drawn the chromosomes from one person in pink and the other person in blue. But you'll notice that the chromosomes are the same length in the two people. They just differ in their colors. In this case, I'm using color to indicate the source of the chromosomes, not the genetic information on the chromosomes. So these two chromosomes will have different versions of the same genes, different alleles of the same loci. These two chromosomes will have completely different genes. And again, here's another representation of chromosomes in a single person. Now I'm only drawing one chromosome using different colors again to indicate that this chromosome came from one person, his mom. This chromosome, this version, came from another person. So what have we done? We've talked about the physical and informational content of chromosomes. We talked about the key regulatory signals that control how chromosomes function, that allow DNA to act as chromosomes. It has to have origins, telomeres, and centromeres. We've introduced other new terminology for dealing with the issues of having different versions of genes. And we've talked about how chromosomes can be represented. Coming up next, we're going to talk about the genes on the chromosomes. I hope to see you there. Hi everyone, welcome back to Useful Genetics. This is Lecture 1J, where we're going to continue our discussion of chromosomes to think about how the genes are arranged on the chromosomes. So what we're going to do is we're going to dive into a chromosome and zoom in until we can resolve a single gene and get a sense of what the gene looks like at a chromosome scale, how much of the gene codes for a protein. And then we'll zoom in on a different gene to see how genes are arranged on the chromosomes. So just to refresh and affirm this very important point from the previous lecture, each of the 23 chromosome types in us, or of the different chromosomes in any organism, has different genes, not just different versions of genes. So we have, the chromosomes are colored here to represent that. So we have two versions of the light green chromosome and two versions of the blue chromosome, and two versions of each of these chromosomes in our cells. And in the whole population, there are many versions of these chromosomes. Now, here's a particular chromosome. This is human chromosome 13. And you see the landmarks that I pointed out earlier, the centromere, and the special end sequences, the telomeres. But what we're going to do now is we're going to focus in on a particular gene. And the gene we're going to look at is the BRCA2 gene. This is one of the genes that's been studied intensively because some versions of this gene cause a greatly elevated risk of breast and ovarian cancer. So this gene is located on chromosome 13, and you can see it's at about 32 million base pairs along the about 114 million base pairs, usually abbreviated megabase chromosome. So we'll zoom in on this, expand this bit to look at just this segment of the chromosome. This is about 2 megabases of chromosome, 2 million base pairs, and the BRCA2 gene occupies this space here. So let's zoom in some more. Here's the 2 megabases segment of the chromosome. We'll zoom in to see the BRCA2 gene. And now this yellow segment is the whole gene. The gene itself, we see, is almost 1 million base pairs long. No, is almost 100 million base pairs long. It's about 90 kilobases, 90,000 base pairs long. And here we have a schematic drawing of the gene in yellow below, but it's kind of funny looking. What this represents is the boxes and the vertical lines represent the exons. And you will remember from an earlier lecture, these are the parts of the protein that code for genes. So that's an exon, that's an exon, that's an exon, that's an exon, that's an exon. There are about 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 15, 16, 18, 19, 20, 21, 22, 23, 24, 25 exons in this gene. But most of the gene isn't exons at all. Most of the gene, like this segment here, is introns. So this is an intron, there, there, there. All of the spaces between the exons are intron sequences, which are cut out and discarded when the mature messenger RNA is synthesized. Now, that's what one gene looks like. More generally, how are genes arranged on the chromosomes? Well, not the way I would arrange them if I was tidying things up. So here's a different chromosome. This is chromosome 20. It's about 62 megabases long. And we're blowing up a segment of it that's about 1, 2, 3, 4, 5, 50 kilobases long. Is that right? No, it's about 500 kilobases long. And now we blow this segment up and ask, well, where are the genes? Okay, there are certainly genes in this segment. There's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 genes in this 500,000 base pairs of DNA sequence. And I'm just highlighting them here. One thing I want to point out, remember when we talked about how one of the things that the promoter does is tell RNA polymerase which strand to use, which determines which direction it goes on the chromosome? Well, here's an example of this. Most of these genes are synthesized off of, it would be the bottom strand in this particular drawing, and they're going this way. But 2 of the genes are synthesized in the opposite direction, which is to say they use the information on the other strand. But the other important thing you'll notice is that there are a lot of spaces in between the genes. So both the order, the direction in which the genes are transcribed, and the spacing of the genes seems very random. It's not neatly organized at all. It doesn't actually need to be neatly organized to work. That's why it's not neatly organized. Now, here's again blowing up a single gene. This is an open reading frame. It's a gene whose function isn't known. It doesn't have a specific name describing its function. It just has a number. Open reading frame number 70 on chromosome 20. And again, in this representation, we see the exons represented as boxes, and the introns represented as spaces joining the boxes. Only the boxed parts code for protein. Only they will be assembled into the final messenger RNA that's translated into protein. So here's a question for you to give you a sense of scale in the chromosome. So chromosome 20 is about 60 million bases long, and it's got about 900 genes. Here's a typical gene, and what the question asks is, if all the genes are like this one, how much of chromosome 20's DNA codes for protein? And to answer this question, you'll need to do a bit of arithmetic. You'll also need to do a bit of estimating. I hope you got that. The right answer is 3%, and this comes from a combination of arithmetic and estimation. What you first have to do is a rough approximation of how much of this gene is actually coding for protein. And just a ballpark estimate, I'd say about 10% of the line is boxes, and the rest is introns. How long is the gene? Well, the gene's probably about 20 kb, and that means that there's approximately 2 kb coding for protein. There's about 900 genes on the chromosome, and if they're all like this one, as we're assuming, that means there's about 1,800 kb, 1.8 million base pairs on this chromosome code for protein. But the chromosome itself is 60 million base pairs, which is 60,000 kb, 60,000 base pairs, and that means that about 3% codes for protein. Now, what we've done, we've dived into the structure of a single gene on a chromosome. We've seen that, the gene's mostly introns, and we've looked at the arrangement of genes over a large scale in the chromosome, seen that most of the chromosome is actually unused space. You'll see this even more dramatically if you look at the chromosome, I think it's chromosome 7 flyover video that's available on the Module 1 page. But despite mostly being introns and unused space, there's still millions of base pairs coding for protein on each of our chromosomes. Now, coming up next, I'm going to take a few minutes to talk about DNA sequencing, just because DNA sequencing has become so important to our understanding of genetics. I hope to see you there.